Fine. Okay, so good afternoon. And initially, I wish to say thank you to all for these opportunities. But before my presentation, I wish to thank you several people here at the BLDE University, especially my good friends, Professor Rosaldas, that organized my visits and is always a friend for now 20 years that we are connected, not only by science, but mainly by hearts, right? Because of that's good, very nice interaction. Thank you for all these efforts. For my second visit to Vijayapura or Bijapur. My first time here was visiting the first time Professor Buzaldas in 2006. So 19 years ago. A lot of good changes in town. Vijayapur is another city, much more modern and a lot of new things in the city, and especially in this university that I am very impressed by all things that we in Luciane, my wife, we saw in the last 2 days. And for this reason that I really wish to thank our Vice Chancellor, Professor Arun Inamadar. Thank you for the warm reception. We are very happy to be here at your university. And some people that were very kind yesterday and today, like Professor Murugesh Patashets, that brought us the idea of the forests, reforestation of the area, which changed the environment, which is a very important issue these days, not only here in Vijayapur, but all the entire world, and the contribution of this university is very important. Also, wish to thank Dr. Prashi Kulkarni, that yesterday we were in the CAMR, that we were very impressed by the facilities, that platform that you help the students, the new generation of investigators in the biomedical fields, in which India is leading the world with the training of a new generation of highest quality experts in different areas of TI and biotech. This is impressive. Sorry that I did something wrong. Sorry. And also, I wish to thank Dr. Lata Malur, that's head of Department of Physiology, that this morning we had a very nice interaction with all the faculties and students and graduate students, and was very effective, our interaction. And also, I wish to thank, on his name, thank all the students that Ali Jawad is following all our steps since we arrived in Belagad airport. So thank you, Ali, for all the supports these days. And all the people that are driving us and the guest house that's making our journey here very, very happy, I should say. This is, in fact, not only the second visit to Vijayapura, but it's my visit number 7 to India since 2004. This visit number 7. Of course, that the COVID break some of our interaction, but even during the COVID, Professor Kuzal does invite me and was online participating in different meetings here in India. So we are very well connected. So thank you all. Okay, so let's move to my talk. And this is our College of Medicine of Ribeirão Preto, is a campus of University of São Paulo. The city of Ribeirão Preto is located 300 kilometers north to the capital, which is named City of São Paulo. So this is a College of Medicine. This is the hospital. This is Children's Hospital. All these facilities are by the hospital. And our basic research building is this old construction. My laboratory is exactly on the second floor here in the back. So if some of you visit us in the future this will be the place. Although these days we are in construction of a new building for the basic research laboratories in this area. And we should be moving to this new facility in the next 2 years. So today, I will try to show you on the next 45 or 50 minutes our current studies in the laboratory involving Neuro-Astrocyte interaction in the brainstem of mice exposed to hypoxia. So I will start this with this cartoon showing you the pathways of the peripheral chemoreflex, which most of you are quite familiar. In case of reduction of the partial pressure of oxygen in the arterial bloods, the peripheral chemoreceptors detect this hypoxia and send afferent information to this area in the brainstem, which is the focus of my presentation today, the nucleus stratus solitarius, which receive information from a large diversity of peripheral afferents. But our focus is mainly the afferents of the peripheral chemoreceptors to the NTS, in which the information is processed in order to connect the ventral aspect of the medulla, the brainstem, in which are located neurons involving generation of the respiratory, as well the autonomic function. In terms of activation of peripheral chemoreceptors in hypoxia, we have the activation of peripheral chemoreceptor, increasing the respiratory frequency and the tidal volume, increasing the ventilation. We have a selective vasoconstriction through increasing sympathetic outflow to the periphery of the vasculature, increase the resistance in order to send the blood flow to the brain and also, of course, to the heart. As well, we have a parasympathetic component that will reduce the rhythms of the heart, reducing the heart rates. So our focus is the NTSC and the synaptic transmission and how it integrates the neural inputs from the peripheral chemoreceptors under condition of hypoxia. So in previous study performed in rats, of course, in my laboratory, we use animal models. We use rodents, rats, and mice. We observed that sustained hypoxia as age increases neuronal excitability and glutamatergic transmission in the NTS of rats, which may contribute to the sympathetic hyperactivity and hypertension. Rats present this pattern of response. When we explore mice, the pattern is quite different. Although they are rodents, their response to hypoxia is quite different. In mice, we observe that sustained hypoxia induces changes in the respiratory pattern, increases the respiratory frequency, and increases the parasympathetic tone to the hearts and pulmonary ventilation, but produces no change no increase, in fact, some reduction in the sympathetic activities. So this was a reason to explore in more details how these rodents, in terms of the synaptic transmission in the NTS, can produce this different pattern of response when compared to other species of rodents, in the case of rats. So here we start to look to the aspects that I will explore in my presentation today. This is the so-called tripartite synapses, excitatory synapses in the NTS. I will be focused my experiments in the presynaptic, postsynaptic, as well how astrocytes can modulate the synaptic transmission. Since astrocytes, one of their major role in the modulation of synaptic transmission is the transporters of glutamate, which remove glutamate from the synaptic clefts in order to preserve the excitability of these postsynaptic neurons in appropriated levels. So This is what we explored, is in tripartite synapses, and the focus was the neuronal activity can be modulated by pre-postsynaptic or astrocytes. Look, all this focus in the synaptic transmission was in order to understand why these rodents, mice, present that pattern of response. So then astrocytes modulate glutamate concentration, as I said, in the synaptic cleft via glutamate transporter, GLT1 englast. So I will use this model in most of my slides. This is the presynaptic, also the astrocytes. Here is the pipette that will do intracellular recording of these neurons. And here, in this case, this electrical stimulation of the presynaptic, the apparatus of these neurons in order to study the presynaptic inputs. So initially, we explore the recording of the activities. Always will be recording the activities of postsynaptic neurons in the NTS. So this is a typical example of the evaluating the AMPA currents. AMPA is one of the subtypes of the glutamate receptors, are AMPA and NMDA receptors, in the case of ionotropic receptors. And this is a typical recording of, in this case, in the voltage clamp, we look for the amplitudes of the currents in neurons from normoxia, the control, and animal neurons from animals submitted to sustained hypoxia. And you can verify that amplitudes of these responses were greater than in control. So something is different in terms of synaptic transmission. If you look to the average of this data, you can see that the amplitudes measured in terms of picoamperes is significantly higher in the group of cells, in group of neurons. Sorry, from mice exposed to sustained hypoxia, excuse me. And the same, if you look to NMDA currents. So NMDA currents, here, you can see that the amplitude of NMDA currents in the NTS neurons of mice exposed to sustained hypoxia was also significantly increased. So, in summary, sustained hypoxia increases the glutamatergic neurotransmission in the NTS of mice. So now we will try to understand in what level of this synaptic transmission occur these changes that allow this increase in the synaptic transmission. So the aim of this first part of my presentation is to investigate the mechanism of increased excitatory transmission in the NTS neurons of mice exposed to sustained hypoxia. So we explore presynaptic, postsynaptic, and the astrocytic modulation. So our main hypothesis, in fact, was that something was different in the astrocytic modulation that should allow increase in the concentration of the transmitter glutamate in the synaptic clefts, which may explain this increasing postsynaptic, the increasing in the excitatory synaptic transmission. But for this, we need to do the experiments and check all these possibilities. And I'll show you at the end that with our hypothesis, we were wrong. There is no chance, as I will show you, in the astrocytic modulation. So in terms of this sustained hypoxia protocol, we expose this C57 black 6J mice to this protocol of sustained hypoxia, reducing the fraction of inspired oxygen to 10% for 24 hours, and the normoxia is maintained in the normal fraction of inspired oxygen. So in these studies, we use Brainstem slices containing the nucleostratus solitarius. With this under microscopy, we identify these cells and we patch these cells. You can see the tip of the pipettes and one of these NTS neurons. We also use multi-electrode array, which is a way to record extracellular. This is intracellular recording. This is extracellular recording of the entire NTS electrical physiological activity. We also use calcium living image and the morphology, especially the morphology of astrocytes in different experimental approaches. So let's first look for the evaluation of the excitability of NTS neurons using patch clamp. So in this is a kind of experiments that we run these experiments to evaluate the excitability. So in this case, we are recording the activity of these postsynaptic neurons, which is the pipettes of patch clamp. This is this electrode stimulation. So with this, we evaluate the number of action potential induced by positive injected currents. And you can see in these recordings that neurons from animals exposed to sustained hypoxia, the number of action potential is increased. If you look here to the summary data, you can see if you increase the currents injected, the intensity of the currents, you can see that the number of action potentials was significantly increased in neurons from animals exposed to sustained hypoxia. So the same is possible with the electrical stimulation. If you induce electrical stimulation, you have the first response in the normoxia neuron. After sustained hypoxia, you can see that the number of action potential is significantly increased. So here is the train of stimuli, 5 electrical stimuli, and then we look for the number of action potential. You can see that the number in reds are sustained hypoxia neurons from animals exposed to sustained hypoxia. So in summary, this data show that in SH, sustained hypoxia increases excitability in the NTS neurons of mice. So using this multi-array recording, recording extracellular for your orientation, this is horizontal, this is anterior, this is posterior. These multi-electrode arrays cover all the NTS. And this analysis show that the overall activity of NTS neurons is significantly increased in the NTS of neurons, of NTS of mice exposed to sustained hypoxia. Therefore, SH increases extracellular activity in the NTS of mice. So all these data are confirming that we have a significant change in the excitability of these neurons in response to hypoxia. So now we evaluated if these changes were due to changes in the presynaptic mechanisms. So the next step was to identify where are these changes, why this excitability is increased. Is due to changes in the presynaptic, postsynaptic, or in the astrocytic modulation, or is involving all these components of the synapse. And for this, here we explore all the parameters related to the presynaptic terminals. including the probability of the release of the transmitters, which can be evaluated by some specific protocols, such as the pair-pulsed ratio, in which we can evaluate if some terminals are releasing more neurotransmitter than in control situation. But in this case, we verified that the presynaptic parameters were not altered by sustained hypoxia. There is no statistical difference in any of this evaluation of the presynaptic terminals. Therefore, sustained hypoxia does not alter presynaptic parameters in the NTS neurons of mice. So it means that that change, that increase in the excitability is not due to changes in the presynaptic terminals. So now we start to look for the astrocytes. That was our major hypothesis to explore these changes in the synaptic transmission. And now we were able, and this is something very exciting in terms of experimental approach, to record the activity of electrical activity of astrocytes. Although they are not excitable cells, they present a difference of potential in between intracellular and extracellular media. So here we are able to identify astrocytes using sulfurrhodamine. You can label the astrocytes, and then we can patch these astrocytes and record their electrical activity. So when we inject currents, this is a characteristic of astrocytes. With this pipette, we can inject these currents inside of the cells. This kind of protocol is controlled, and you can increase the intensity of currents, and you see a linear changes in the potential, membrane potential, but it never generates action potential. It is not excitable. It does not present any sodium channel, so it does not depolarize, but you can induce change in the membrane potential. This is a good way to characterize astrocytes, and we are very happy that we were able to record the activities, because we are sure that we are really working with astrocytes. And then we verified that the membrane, the resistant membrane properties of astrocytes was similar between control and sustained hypoxia group, showing that sustained hypoxia does not alter the resting membrane potential of NTS astrocytes. Of course, this was not enough to rule out the possible involvement of astrocytes in the change of the synaptic transmission. Then we start to explore the activity of the glutamate transporters. Now look, our pipettes is here in the astrocytes, and we do a series of protocols in which we use drugs such as DNQX, AP5 to block all glutamate receptors, TTX, tetrodotoxin, to block the activity of neurons, because it blocks sodium channels. And we activate these transporters with the aspartates. And this, when we look to the currents induced by the aspartates in these transporters of glutamates in astrocytes, in control, normoxia, and animals, and neurons from animals exposed to sustained hypoxia. And we observe no difference in between the currents of the transport of glutamates. This is the way to evaluate the transport of glutamates in astrocytes, and it was not affected by sustained hypoxia. So the summary of this data: sustained hypoxia does not alter the glutamate transporter activity in NTS astrocytes. So it means that it does not have an increase in glutamate's concentration in the synaptic clefts. So this is not the cause of that increase in the synaptic transmission. So the next was to evaluate the intracellular calcium in the astrocytes. For this, we use fluorophore as a marker for calcium and sulforhodamine to identify the cells that are astrocytes. And then using a multiphoton microscopy, we evaluated, in this case, is a control, artificial cerebral spinal fluid perfusion. And here we have a perfusion with AMPA agonists. AMPA agonists of AMPA receptors, of course. And again, we observed that the AMPA maximal response in neurons of normoxia and sustained hypoxia animals were not different. So again, sustained hypoxia does not alter the intracellular calcium mobilization. The intracellular calcium mobilization evaluated by the fluorophore, which is the marker for the intracellular calcium evaluation through this approach. So the next look, this was our hypothesis, and we are trying, we were working hard to verify if the astrocyte was really involved in this increase in the synaptic transmission. For this reason, that we also evaluate the morphology of astrocytes using GFAP immunohistochemistry, as well transgenic animals expressing the GFAP and GFAP mice in order to evaluate the morphology of astrocytes. And here we analyze the area of GFAP using conventional immunohistochemistry, as well in these transgenic mice exposed to sustained hypoxia. We evaluate the GFAP green fluorescence protein in astrocytes from normoxia and sustained hypoxia, as well the cell process, because look, the contact of the astrocytes with the synaptic transmission is not due to the main body of the astrocytes. In fact, the process of astrocytes that send the projections to the synaptic transmission, and it is modulated, in fact, by the process. And also the axis or the distribution of this process from the astrocytes. And we observe, again, no significant difference in the morphology of astrocytes. So all these data are showing that astrocytes are not playing any major role in that increasing the excitability of NTS neurons in response to sustained hypoxia. So let's look to the postsynaptic aspects of these tripartite synapses. And here we evaluated the amplitude of the AMPA currents, recording the pipette is in the postsynaptic neurons. We recorded the activity of these neurons in the presence of TTX. FAC is to inhibit the activity of these astrocytes, is a metabolic inhibitor of astrocytes. And then we use AMPA to activate its AMPA receptors. So we block the neurons with tetrodotoxin to avoid any excitatory inputs other than the perfusion of the AMPA agonists. And look, when we apply in the beds this AMPA agonists, we compare the amplitudes of these currents, and then we observe a significant increase in the AMPA currents in these postsynaptic neurons. The same was observed with respect to the NMDA currents. So in fact, what we observed that sustained hypoxia increased AMPA and NMDA postsynaptic currents in the NTS neurons. So the problem, I should not say the word problem. These animals, they were exposed, in fact, to a problem that was sustained hypoxia, and their physiological response, which is very smart, as all the physiology is to adjust the central nervous system to this new challenge. And the response of the central nervous system, in this case, in the NTS, is produce adjustments in the postsynaptic level, increasing the excitability. Probably we do not have yet data about the changes in the characteristics, the kinetics of these channels that are opened by activation of AMPA and NMDA receptors, which they are itself channels. Through them goes the currents of sodium that depolarize these neurons. So, but this is the major hypothesis that now we are working on, that is changes in the kinetics of these ionic channels that may explain the increase in the excitability of these neurons. So in summary of this part one, fortunately, I have time for the part two. The summary is the following: sustained hypoxia increases the excitatory synaptic transmission in neuronal excitability and in the NTS of mice, due to increase in AMPA and NMDA postsynaptic currents, other than presynaptic parameters, which were not altered, neither, or astrocytes-related mechanisms. So, in conclusion of these parts, the observed changes in the modulation of NTS synaptic transmission in response to sustained hypoxia in mice are different. So this is the first observation in relation to those previously described in rats, not only in our laboratory, but in several laboratories. And also the increase in the postsynaptic glutamatergic currents in NTS of mice is consistent with the autonomic and respiratory profile observing these rodents after sustained hypoxia. Mice, they, as I said in the second, third slides, they responded different of rats in relation to sustained hypoxia. They have a reduction in the sympathetic tone, increase in the parasympathetic, they increase in ventilation. More recently, you have data regarding the body temperature. They reduce the metabolism, they reducing the body temperature. This is something that we must pay attention to, the metabolism of this. Any animal submitted to hypoxia, the body temperature is a critical index of the metabolic activity. And the mice behave very well. They reduce, in the first hours of hypoxia, they reduce the temperature in 3-4 degrees Celsius. And they stay very quiet. So they know that they are being threatened by the hypoxia. And everything is, in our view, in mice, they are responding in order to lower the oxygen consumption, which is low due to the sustained hypoxia, lowering the metabolism and producing autonomic and respiratory response, reducing the heart rates, reducing oxygen consumption, increasing the respiratory ventilation in order to increase the pressure of oxygen in the arterial bloods. So mice are much well prepared by the nature to face hypoxia than rats, and for sure than human beings, no doubt. Therefore, this is a question for our later discussion. Are mice the good model for us to study hypoxia? Depends of your questions and your view, but they are very, very efficient, and we must learn with them. Okay, so the second part, I have more 20 minutes. Mr. Vice Chancellor, 20 minutes? 15 minutes? Okay, I will try to, yes, I will be able to do that. Okay, in the part 2, we are looking at the same time in the laboratory, we are exploring the role of astrocytes under hypoxic condition. So the question here is, what is the role of adenosine receptors, A2A and A2B, in neurons and astrocytes intracellular calcium mobilization in mice exposed to SH? So the question here is astrocytic modulation of neuron activities and the role of adenosine receptors. Why adenosine? Adenosine is released in circumstances of hypoxia. So in any case of metabolic change, reduction of oxygen or increasing in CO2, we have a release of adenosine in different areas in the brain, and specifically in this area in the brainstem, NTS, as well in this ventral medulla. And adenosine play a critical role in these astrocytes. And our question was in relation to the increase in the baseline respiratory pattern in response to sustained hypoxia, increased the respiration. In the previous data, we used C57 black 6J mice, normal mice. Here we introduce the knockout mice for A2A receptors in order to verify in what, since we have a previous evidence that A2A receptors can modulate the respiratory rhythm, the respiratory frequency. Excuse me. I have oxygen enough, but I need some water. I am not yet under hypoxia, right, Professor Das? Yeah. Okay, so in this part of this, I will use whole animal that we are conducting some experiments in vivo, in the working heart brainstem preparation, as well in vitro PET clamp, as well calcium living image. So here we use, as I said, male knockout mice for A2A receptors, and the same protocol of hypoxia. And when we recorded these animals, whole animals, awake, freely moving mice using plethysmography, we observed after sustaining hypoxia a large increase in the respiratory frequency. So a significant increase, their respiratory frequency is very high in mice, nearby 200 cycles per minute, and it increased to approximately 260, 270 cycles per minute. There's a very high frequency. It's very difficult to be small animals in terms of balance of the temperature, the ratio of their body's super area by the body mass is not advantage to be a small animals. They expend much more energy, oxygen, to keep the body temperature. It's better to be an elephant than a mice in order to keep the control of body temperature. So these knockout mice, A2A, they present a large increase in their baseline respiratory frequency. So it means that in some way, A2A receptors are in fact involved in the modulation of the respiratory frequency. After sustaining hypoxia, they present no major increase in the respiratory frequency. In fact, they present a reduction because their baseline is higher than wild-type mice after sustained hypoxia. So the idea is to understand how adenosine and these A2A and A2B receptors regulated the respiratory frequency. So adenosine A2A receptors play a major role in modulation of respiratory frequency in awake mice. So this is recording from the working heart brainstem preparation. This is normoxia, and this is sustained hypoxia. This is just for your orientation. This is the recording of the phrenic nerve activity, which is the index of respiratory frequency, of course. In knockout mice, you can see the frequency of the baseline frequency of the phrenic nerve discharge is much higher than in normoxia, in the control animals. When we increase producing hypercapnia, increase the activating the central chemoreceptors, we have also different pattern of response. So in this case, when you compare, this is knockouts, data from knockout mice, control and sustained hypoxia. If you compare the phrenic nerve discharge, it's completely different in relation to the control. And the responses to hypercapnia, 7 and the maximum 10% of CO2, they present a reduction. So in some way, A2A receptors is part of the regulation of the respiratory frequency. In these animals, in the baseline, they are really regulating the respiratory frequency. So in the control of knockouts, you can see the difference. So the major problem here is not the hypoxia, is the lack of A2A receptors. So the baseline phrenic nerve discharge in knockouts A2A mice was higher than in the wild-type mice, in this case, BLDE. So here we test the record the phrenic nerve discharge in response to central or peripheral chemoreceptor activation in the wild-type and knockout mice. And we verify, just have a look in this case, that the responses to central chemoreceptors is completely blunted in knockout mice in response to hypercapnia, as well the response to peripheral chemoreceptor activation. So when we activate the peripheral chemoreceptors in normal animals, we have a large increase in the respiratory frequency, in the phrenic nerve discharge, which is not observed in animals knockouts or A2A receptors. So the increase in the phrenic nerve discharge in response to central hypercapnia or peripheral we inject cyanide chemoreceptor activation was blunted in A2A knockout mice. So the next experiments that I will show you, the specific aims were to evaluate the effect of sustained hypoxia on the intracellular calcium mobilization during the glutamatergic transmission in the NTS of mice, as well to evaluate the role of A2A and A2B receptors in the intracellular calcium mobilization during the glutamatergic transmission in the NTS of mice after sustained hypoxia. For this, we use mainly imaging of astrocytes and neurons. As I mentioned before, fluorophore is the marker for calcium, intracellular calcium, and sulforhodamine 101 to identify astrocytes. We use the fluoview multiphoton laser scanning microscope. And here we use the AMPA perfusion, relative changes in the fluorophore fluorescence versus time determined by image J. That was the way to analyze. And then we look for the number of regions of interest, the so-called ROIs, and the number of slices and the number of mice. and we apply AMPA to produce the maximal response. In this case, we look to the NTS neurons. The question was, is the increased AMPA currents in the NTS neurons of mice after sustained hypoxia associated to change in fluorophore fluorescence, I mean intracellular calcium mobilization in neurons? No, there is no change in the intracellular calcium mobilization in the NTS neurons after sustained hypoxia. So fluorophore fluorescence in the NTS neurons is not affected by sustained hypoxia. So the next question was, are the changes in the fluorophore fluorescence in the NTS neurons of the wild-type mice after sustained hypoxia dependent of A2A receptors? This is in neurons. So this is the wild-type normoxia sustained hypoxia, knockouts A2A. There is no change in the AMPA maximal response. And then we also use the traditional pharmacology using A2A receptor antagonists, so-called SCH442416, and again, produce no change in the intracellular calcium mobilization in the NTS neurons. So A2A receptors are not related to change in fluorophore fluorescence in the NTS neurons after sustaining hypoxia. So the next question are several questions. One series of experiments bring the next question. Is fluorophore fluorescence or intracellular calcium mobilization altered in the NTS astrocytes of mice, especially in the wild type after sustained hypoxia? Sorry. Then, I think that I went back, sorry. This was in NTSC, sorry, go back in. This fluorophore, this one, yeah. This is fluorophore altered in the, I'm doing something wrong here. In the astrocytes of mice wild type, produce no change in the fluorophore fluorescence in the NTS astrocytes. This was the experiments with the pharmacology, block of A2 are involved. This now is the fluorescence in the NTS astrocytes of wild type in the knockout mice. Yes, in this case, we observe a major change in the astrocytes, intracellular calcium mobilization after sustained hypoxia in knockout mice. This was a significant increase. As well, when you use the A2A receptor antagonist. In fact, these knockouts mice or wild type, when you perfuse with this A2A receptor antagonist, we observe an increase in the calcium mobilization. So the lack of A2A receptors are involved, in fact, in the increased fluorophore fluorescence in the NTS astrocytes after sustained hypoxia. So in fact, astrocytes represent some change in response to sustaining the hypoxia. What is the functional implication of this is one important question. So here we look to these effects. The question was, is the A2B receptor activation related to the increased calcium concentration in the NTS astrocytes from A2A knockout mice after sustained hypoxia? Yes, when you use this, and this is neurons, the antagonism of A2B receptors block the increase of fluorophore in the NTS astrocytes of NTS neurons. If you remember the previous data, sustained hypoxia increases the calcium mobilization, especially in knockout mice. Here, when we use A2B receptor antagonist, that increase was abolished. So it means that the increase in calcium mobilization is due to activation of A2B receptors. Since the antagonism of A2B receptors in the wild type, we, especially in knockouts A2A receptors, we blockade the increase in fluorescence in the NTS astrocytes after sustained hypoxia. So another question was, is the increase in fluorescence in the NTS from knockout mice for A2A receptors after sustained hypoxia linked to neuro-astrocyte interaction? This is data that I presented before. Knockout mice for A2A present an increase in intracellular calcium mobilization. Is this increase related in some way to the neuronal activities? How you test this? Using tetrodotoxin, we block the neuronal activities, and that increase in calcium mobilization was abolished. So this calcium mobilization in the astrocytes in the NTS in response to sustained hypoxia are in fact dependent of the activity of neurons, since when we blocked the neuronal activities, that increase was abolished. Another point another question, is the increase in the fluorescence in the NTS from A2A knockout mice after sustained hypoxia linked to neuron-astrocyte interaction? The response, as I mentioned before, yes. The modulation of A2A receptors of increased fluorescence in the NTS astrocytes is dependent of neuronal activities. So what is the source of this calcium that is mobilized in the astrocytes of the NTS in these knockout mice exposed to sustained hypoxia? If you run these experiments in a calcium-free solution, we verified that increase was abolished. So this calcium came from extracellular store, is not intracellular calcium mobilization. So the increased calcium mobilization in the NTS astrocytes from these knockout mice is dependent of extracellular calcium concentration. So in summary of this second part, in response to sustained hypoxia, the antagonism of adenosine A2A receptors, the intracellular calcium mobilization in NTS astrocytes increases due to activation of A2B receptors. And this is a problem in this kind of model. Knockout mice for A2A, you are changing the balance, which is in normal animals, A2A and A2B receptors are modulating this calcium mobilization in astrocytes. But in knockout mice, the lack of these A2A receptors occur a predominance of A2B receptors, which increase the calcium mobilization. This effect of increasing calcium mobilization is dependent of neuronal activities and extracellular calcium influx. These findings contribute to explain, at least in parts, the observed changes in the respiratory frequency modulation and the enhancement of AMPA postsynaptic currents in knockout mice, in the NTS neurons of knockout mice after sustained hypoxia. Whether or not this increase in the astrocytes intracellular calcium mobilization contributes to provide energy, specifically lactates, to the neuronal activities is still matter that is currently under investigation in our laboratory. So as I showed in the first part of my presentation, as well in this second part, astrocytes are not directly involved in the modulation or the change in the excitability of synaptic transmission after sustained hypoxia. But these data are suggesting that lactates should play a critical role in providing support to the neuronal activity, specifically increasing the lactates, the source of energy to the activity of neurons. And this is a very new field to be explored in terms of how the astrocytes provides energy to neurons, specifically under circumstances of a challenge such as hypoxia, sustained intermittent of any kind of hypoxia that may bring risk to the whole entire activity of the brain, and specifically these areas in the brainstem, which are critical for the autonomic and respiratory response when the animal or the human beings face such a risk situation such as hypoxia. With this, I just wish to finish and mention the acknowledgements of people in my laboratory. Dr. Daniela Cortes Mendonça is a researcher, investigator in my laboratory. Professor Davi Moraes was a faculty in our department, and he moved to the city of São Paulo, to the same university. Juliana, Carla are graduate students in my lab. Ludmila was a postdoc. Luis Henrique also a postdoc, and Mikhail finished his thesis 1 month ago. Also collaboration of Professor Sylvia Rudzinski of University of Alfenas, and some collaboration with Professor David Klein and Protopio Barcelos at the University of Missouri in the United States. And the important financial supports by our state foundation and 2 federal agents in Brazil, the federal government that supports our projects and our laboratory with fellowship for the personnel and equipments and all facilities in our departments and laboratory. And this is the site of my laboratory for those that wish more details about our publication. And this is my email that I will be more than happy to ask to receive any comments and questions that I will be very happy to interact with you all. So thank you very much for your attention. And I finish a little bit later than expected, but if you have time for a question, I will be more than happy. Thank you. Thank you.
I'm sure of you are breathing and awake. Well, that's all because of the neuron and astrocyte interaction that is all conscious and breathing.
Yeah, that's right. Good.
Well, Professor Benedicto Masado, really it was I would put it as a scholarly talk, simply because we have people from physiology and other science and technology people, and they work in people from physiology, especially the vascular laboratory.
That's good.
And what I could understand as a non-physiologist in going through your talk, the very basic thing is putting a research hypothesis and then question after question after every experiment and the results. And result need not be always yes. If negative also, you have to question again why it came as negative. Probably with these things, this is how things should work in physiology or the basic science. And we being the clinicians probably only observables, like we do observe things, report or conclude something. That's why Just yesterday, the Nobel Prize was announced. It is named as physiology and medicine, not just chemistry.
Yeah, that's right. Yeah,
They have thought about these things, and I wish our colleagues here take a leaf out of these experimental things, how it should go about. And then there is a lot to come from the basic science, and then only the university becomes really university. Otherwise, just we can give degrees to people. And I was astonished to read. It's true also. The Cambridge University announced yesterday that the person who got the alumni of that university, and it is the 186th Nobel laureate from Cambridge University. Well, we will not reach those. You never know. Maybe after 50 years, 100 years, the seeds should be sown from at this stage. Dream, dream something. Probably you start walking along that. And unless you keep walking, milestones should not be crossed. So we cannot reach the goal. Anyway, it was just amazing, sir. If there are any questions from my physiology colleagues, I'll be more than happy to pass on the question.
Sure. Yes, it's my pleasure to try to clarify some issue that I was very fast in some of these slides, and I'll be more than happy to.
Interesting.
Please.
As per your part one of the experiment, there was no role of astrocytes and presynaptic neurons in altering the synaptic neuronal activity. And you also mentioned that there was a significant change in the body temperature of the animal whenever the animal is exposed. So I was thinking in those lines whether the temperature is altering the kinetics of the receptors and the channels.
Yes, this is, of course, this is something that we must take in
Suppose we keep the body temperature constant, expose the animal to hypoxia, and then study the activity along the channels. Will it significantly affect the channel activity, movement of the ions, opening and closing of the channels? Because that is one important parameter which can influence the whole setup.
I agree completely. You are absolutely right. And this is a very important question for all us physiologists or involved in the medical or biomedical fields. Look, we are a kind of physiologists. I consider myself an integrative physiologist. And look, we are exploring ionic channels in the postsynaptic neurons in the mice. If I do not have a big picture about the integrative physiology, the control of autonomic, respiratory, temperature, all these parameters that are essential for the cell function in the ideal environments, I should be missing so many information. For example, the temperature was something that was after we recorded carefully with very small sensor, intra-abdominal sensor that we exposed these animals to 24 hours of hypoxia is a remote control of the body temperature. When we verify this huge fall in temperature, 4 degrees isn't amazing. So I said, of course, it's obvious that the animal should be present some change in temperature. Why we are not thinking about this before? So this is an example that we physiologists must have the big picture and try to understand how the system works, and not only how the ionic channel or the AMPA or NMDA receptors are more or less active, because I could be losing essential information. And the system is so complex and so well organized that any change in the body temperature, of course, that the activity of this entire system is different. You're right. If you warm up these animals during the hypoxia and keep their body temperature near the normal level, this data could be completely different. Oh, no doubt. I agree completely. But this is a kind of situation that we must be aware and not look for the receptor without any other factor that can be affecting the phenomena that I am looking for. And this is what makes us physiologists specific biomedical professionals that look for the entire the integrative system and how it works, taking account all these possible genes. But this is a very good idea to keep the temperature and try to see how these cells respond. Yeah, no doubts. You're absolutely right.
Thank you. Any other questions from anybody? Okay, with this, I will take opportunity to first thank from our university that Professor Benedito Machado has come all the way from Brazil, Sao Paulo, very beautiful country, and
you know how far is Sao Paulo because you was there recently.
3 months back, I was there in Sao Paulo, and then I went to another viewer also, so we were just discussing with them.
Yeah.
Very good hospitality, and 2 words I learned from Brazil, which are there in India too, because Portuguese is the language we speak, and vada pav, what do you call this? Pav is the bread. So I put in bottles also pav, different kinds of pav. So all these pav, and then the potato, potatino, potato. So anyway, it was a nice place. If anybody gets chance to visit Brazil,
You'll be more than welcome, for sure. Yeah.
and we have with us Professor Madam Luciani, the better half of our Professor Machado. She's a pharmacologist also. Probably that's why there were some things where pharmacology was also discussed.
That's right. In some way, I'm influenced by the pharmacologist at home. Yeah.
Yes.
Great, thank you.
Thank you, everybody. I, on behalf of University, once again thank Professor Machado for being here and delivering his lecture. I'm sure, sir, your expertise must have given a deeper understanding of the subject among the audience. Thank you again for flying all the way from Brazil to India and visit BLDE University. It's indeed our great pleasure to have our Honorable Vice Chancellor, Dr. Arunanand sir, to chair this session. Thank you for graciously accepting our invitation to chair the session, sir. Despite we all know his busy schedule, he is here. Thank you so much. I would also take this opportunity to thank Professor Das. I think the most pivotal person to get Professor Machado to BLDE University. Thank you so much, sir. I also thank all the faculty, postgraduate and undergraduate students who have come here with full enthusiasm to understand, research, and to know about this particular subject. Thank you, everybody. I thank Department of IT also for always being supportive to all the IT activities that we conduct. Thank you. Thank you.